Okay. Okay, so starting over again. So we're going to start by labeling the parts that we've got here. Here is the stroma. Here is the lumen. This is a thylakoid. So we're going to outline the light-dependent reactions, or how this all happens, the pathway in photosynthesis. So this is photosystem 1. Here is photosystem 2, sorry. And here is photosystem 1. And here is our ATP synthase, or ATPase. So we're going to start out with a photon of light coming down into the chlorophyll, or into the chloroplast, the organelle that's in a plant cell that's going to condition these photons to be at the right wavelength. Once they're at the right wavelength, they power up enough energy to break apart a water molecule. Once they break apart that water molecule, they get an electron from it, oxygen, and protons, right? So we've got hydrogens and oxygens that make water. And now we're going to break them apart right here. Taking the energy from that photon, we're going to split that apart. Oxygen is a waste, so that's just going to leave. But these protons are going to keep building up in the cell, and building up, or in the lumen of the thylakoid, keep building up and making protons. So once we've excited this electron, it gets to a higher energy and gets passed off then. And we call this the electron transport chain, ETC, the electron transport chain. So this electron gets passed on. As it goes through this middle protein, it causes enough energy for us, and I'm going to use a different color, for us to pull protons down through this protein. So that energy from that electron is used to pull protons into the thylakoid. So we're adding more and more protons again every time an electron comes through. Now this electron will get passed eventually over to photosystem 1. Now at photosystem 1, again, a photon of light comes down, provides enough energy to excite this electron. Now that electron can be passed off to the next protein, where it is going to use its energy to power up an NADP plus into an NADPH. So this one is now fully loaded with electrons and ready to go over to the Calvin cycle. Now that electron also, remember, has the option, it can keep circulating. It can come back through this protein and keep pulling protons down and take this path, or it can come through and take that path. At that point, the electrons can go either way. So now we've made the NADPH that we need for the Calvin cycle. Now we're going to take these built-up protons in the lumen, and they're going to make their way over to this ATP synthase. And so since they're highly concentrated on the inside, that means that they're going to have a lower concentration on the outside. They're going to go down their concentration gradient and get pushed out of the thylakoid, out into the stroma. So when they get pushed through, every time one of these protons gets pushed through, it causes this to spin. And when that spins, it's going to take an ADP, adenosine diphosphate, and turn it into a powered up ATP molecule. It's going to tack on another phosphate group. So every time one of these hydrogens, one of these protons gets pushed through that protein, it makes the top spin, gives enough energy for that ATP molecule to get powered up. So now using photons, water, electrons, we've now made ADP into ATP and NADP plus into NADPH. And now those two are going to make their way into the Calvin cycle to start making glucose for the cell.